Hi, my name's Neve Hannigan and I am co-director of the BRI, which is just a student-led research institute which we have at Life Sciences UTC. And when we've been given the SEM, we sort of took it amongst ourselves to create a little group who are interested in using it and to begin some research projects. So I've become sort of administrator along with some other people who are going to be speaking today of running the SEM, which has been quite a nice little opportunity. So basically, as you can see on the presentation, our title will be creating and running a student-led SEM program to promote independent learning and research, which basically just means it's just going to be an explanation of how we've sort of learned how to use the SEM and what we've used, how we've used it in our projects and how we've helped other people use and do projects with their own samples and things like that. So below you can see some of the images that we've obtained from some of the year 12 pieces of projects. So we can see a mouse scapula, which is just the shoulder blade of a mouse. And you can see some artifacts on it, and you can see the paws and dimensions and things. And the second one along is just one of our projects was figuring out if there are any microbes or things on surfaces. Obviously, due to the current climate, people are a lot more conscious of cleanliness. So a couple of girls wanted to study that, and you can see what we think is a fungal colony on a keyboard of a computer, but we're not entirely sure. And the last one is a locust eye, which is by Jasmine, who will be speaking in shortly, which is quite interesting when you think about it. Below as well, you can just see it says 15 kilovolts, which is just the acceleration of the electrons that are firing towards a sample, which is just extreme. So it's amazing that we can get the images, really. So yeah, I'll just hand you over to Jasmine and she's going to talk about what we've kind of developed and how we've obtained some new skills.
Hi, I'm Jasmine Spencer. I'm part of the technical centres focusing mostly on model organisms. When we got the SEM, we've been trained to use it by Miss Linkman and Dr. Dyer. This is to allow everyone to have the chance to use the SEM in the school and allow everyone to see their own images. We've been able to collect quite a few unique and interesting samples. For example, the hoverfly wing shown on the pictures and tarantula foot. We've also prepared the samples. To do this, we used the carbon sticky discs and metal stubs. We also had to wear gloves while doing this to prevent them from being contaminated. We've also learned how to calibrate the SEM for easy use independently so that we can set up a facility for other students to use. We know how to do this by closing the machine to create a vacuum essential for the electrons to pass through. Also, we have been trained to use the software to ensure a high standard of images are collected for all of the students. To do this, we've used different settings, which include the increasing and decreasing the magnification and the brightness and the contrast and the different modes to get different magnifications. The other images on the slide include the maggot head, which is for a student's HPQ, studying the life cycle of arthropods to determine different stages of decomposition, and also the mealworm, for a Year 9 project surrounding the digestion of plastic waste.
Hello, my name is Mohammed, and I'm part of the technical model organisms in the BRI, which has been introduced by the first student, and I take care of the fish in the school. And my EPQ is sided with the aquaponics, which is the cycle between fish waste and plant production. And I've been using this opportunity with the electron microscope to further my research into fish, which I would like to next look at the scales, at the age rings to see the ages to differentiate between them and see the effect it have on plants production. And these three groups you see in front of you are also people who have been trying to use the electron microscope to further their own project. First of all is Isaac, whose project is enzyme transglutaminase, which is used as a wound glue for the muscles, which they want to make a part of the first aid, and they're using pig muscle as a model. And due to the light microscope not being able to provide them with enough help to further it, they've sent it to the University of Liverpool because we had not received the SCM yet. So they want to send it to University of Liverpool to check and see how much of an effect it has on the muscles and how much it would glue it back on. As you can see in the picture are the muscles and what they coped with. And there's Josie and Julia who are investigating the microbes on different surfaces due to the current climate and the coronavirus, which is fairly effective against the virus, and it's spreading against every surface that we are touching, and as we are trying to protect ourselves. For example, the computer keyboards, the door handles, sinks, and light switches. And then there's the Year 9, the Real Meal Group, which they've named themselves and are a part of a competition which they've reached the finals, investigating the potent of a mealworm larvae digesting the plastic waste, which is one of the major things that's happening in the world right now, affecting everyone, as you may see with the things that have been going on. And they're trying to investigate the mealworm's guts to see the plastic waste and how it's processed in its guts. Thank you.
My name's Charlotte. I'm part of the technical centres team in charge of the SEM. And so obviously this has been an amazing opportunity for both learning and also gaining experience in terms of university. So my year 12, we've been able to both use this for our EPQs, as mentioned previously by Mo. We've been able to start our own independent research projects. This has been really interesting. Also, it's inspired other EPQs. And as well as this, part of the BRI have been able to gain skills using the SEM to help the younger years, like year 10, the Ignite group and the PVL group. That's been really that's been a really good experience for us in terms of gaining those skills, for like writing that down in our personal statements for university. And it's been great for the year 10s because they've got to observe samples and do their own projects.
Thanks for listening. Have you got any questions?